Hi, I'm Megan Grove, and today in this video we're going to be talking about a few in silico prediction tools that we use to computationally predict the effect of a variant on a protein or a gene. So for the purposes of today's video, we're only going to be briefly touching on what in silico programs use to generate their predictions, and then we're going to actually show how you input a variant into three commonly used tools: polyphen, MutationTaster, and SIFT. So for a more comprehensive list of the different tools that are out there, because there are many, and how to really interpret that data, I want to point you to the American College of Medical Genetics and Genomics guidelines on sequence variant interpretation, published in Genetics and Medicine in spring 2015, because that has a very helpful list of many of the algorithms I have showing right here. Table 2 lists many of the missense and splice site prediction tools that are out there, where to access them, and then what underlying data do they use to generate their predictions. And then they also incorporate how to interpret this data in the context of the rest of your evidence that you're reviewing on a particular variant in your assessment of whether or not that variant is pathogenic or benign. So I definitely encourage you to take a look at that resource for a more comprehensive overview of in silico tools. So to start, I'm going to, as I mentioned, take you through a couple example variants through polyphen, MutationTaster, and SIFT. And just as a general overview, these in silico tools, our goal is to use these to help us with interpreting sequence variants, and as I mentioned, those are mainly missense and splice predictions. And so these different tools, while the different algorithms can look different, the inputs can look different, overall, a lot of these tools use the same underlying assumptions and data to generate their predictions. So these include things like the variant change itself, the location of that variant change, so is it at a position, either a nucleotide or a residue that's evolutionarily conserved? Is it within a known and annotated protein domain? And what's the biochemical nature of the affected amino acid residues? All of those types of data are used by many of these prediction tools. And so consequently, we can't use these different tools as independent pieces of evidence. So if we looked in polyphen, mutation, and SIFT, or MutationTaster and SIFT, for instance, and they all said disease-causing or damaging, we can't count that as three pieces of evidence. And instead, the American College of Medical Genetics and Genomics in their guideline recommends that you do use multiple tools when you're trying to obtain a computational prediction of the effect of your variant, and only if those tools agree can you count that as a piece of supporting evidence. So in general, these tools can be helpful when assessing a variant, but they themselves should never be used as a sole piece of evidence for your assertion of a variant, and they can't determine or rule out pathogenicity of a variant. Okay, so that's just kind of a high-level overview, and as I mentioned, for more information, I would refer you to that guideline document. Okay, so we're going to first start with KCNQ1 example missense variant. And we're going to take you through polyphen first. So this is a KCNQ1 missense variant, tyrosine to cysteine at position 315. So for polyphen, as I mentioned, it is a missense variant predictor. And the input for that is the protein nomenclature describing your variant, or the P-dot nomenclature. So here is the website for polyphen, but you can also get there just by Googling polyphen. It's one of the top hits. And this is the polyphen home screen. So basically what you would input is your gene name, so KCNQ1 in this case. If you have your particular transcript or KCNQ1 transcript that your laboratory provides you with, you can obtain the associated ensemble transcript. So the lab will most often provide you with this RefSeq transcript, this NM number. And if you look in, there are many resources where you can do this, but if you look in NCBI gene at the KCNQ1 gene, you can pull down to the NCBI reference sequence section. And then as you'll see, I've highlighted here the RefSeq transcript that was provided to me by the lab. And then I can see related transcripts, and the related ensemble transcript is right here. So if I were interested in querying polyphen based on a particular transcript, I could highlight this and input that instead of the gene name. If you have remaining questions about alternative transcripts and you haven't already watched the how-to video on variant nomenclature and finding out more information on alternative transcripts, I'll point you there for additional information. Okay, so once you put in your gene name, then you need to put in your protein or amino acid position that's affected. So here, that would be this 315. And then you need to put in the original amino acid and then your new amino acid seen in this substitution. So in this case, we're going from a tyrosine to a cysteine. And as you'll note, the amino acids are indicated by the single letter representation of the amino acid, but if you hover over it, you can see the full name. So we are going from tyrosine, which is a Y, to cysteine in this particular KCNQ1 missense variant. Then you hit Submit Query, and it's going to take you to this progress page. As you can see, there's a completed section and a pending section. So I've already been querying variants today, so that's actually showing the results from my previous searches. But then there's this one pending running query, which is the one that we just submitted. Sometimes if you look in this load section, it will give you a sense of how many jobs are ahead of you. And so if the load is light, we're not going to have to wait very long to get our polyphen prediction. But if that load is heavy, it could take a few minutes, so you might actually want to go do something else and then come back. So I'll hit refresh, and then you can see that the variant that I just submitted, the results are ready. If there was any error in the input that you provided on the original home page, then that would be indicated right here. So in this case, I'll click on the results. As a starting place, I like to just double check that I'm really looking at the same variant. So I know that we were at position 315 in the KCNQ1 gene and that we're going from a tyrosine to a cysteine, so we're all good there. It's just good to double check that you're actually looking at the right variant. And you'll see it defaults to show you this HumDiv model predicting probably damaging. But actually for looking at human variation that is thought to be the underlying cause of Mendelian disease, they recommend that you use the HumVar output, which you actually need to expand on that results page. So as you'll see here, in this case, both are agreeing with each other. They're predicting probably damaging. And for polyphen, that is the equivalent of polyphen's highest level of saying this variant is predicted to damage the typical protein gene function. The other possible outputs that you could see from polyphen are possibly damaging and benign. And then it tries to give you an indication of confidence or a score underlying that prediction. I find in their About section, you can find FAQs and more information describing the score. But generally, the closer to 1, the more confident that they are in that prediction. And in this case, given HumDiv and HumVar were the same, it's okay if you had accidentally just looked here, but that's not always the case that there'll be an agreement. So again, just make sure you're always focusing on the polyphen HumVar model output. Okay, so that is polyphen. So for this particular KCNQ1 variant, it predicted probably damaging. So we're going to take the same variant and put it through SIFT next. So SIFT is another missense prediction program. Here's the website here. You can also, again, Google search to get to the home page. And a key difference about SIFT is while it is also a missense variant prediction program, the input is different. So instead of inputting the PDOT or protein location nomenclature, you actually have to input the genomic position. So not necessarily every lab will be providing you the genomic position of your variant. So you might, in some cases, need to look that up yourself. I'm not going to show you how to look that up in this video, but if you have questions about that, I'll point you to the variant nomenclature and genomic position how-to video, because we show you exactly how to do that if you're given protein and cDNA nomenclature for a variant, but not genomic position. So for this particular KCNQ1 variant, I've already went ahead and pulled the genomic position. So it's chromosome 11, then here's the GDOT or genomic position, and then here is your single nucleotide variant. So let's go to the SIFT homepage. So if you Google search the SIFT page, you'll come across this homepage, and then you would click on the SIFT human SNP. And then basically your input would be in this box right here. So I find it very helpful to first double check you're using the same human reference sequence or the human reference build that the laboratory used. Right now, that's pretty much universally this HG19. but just something to keep in mind. Make sure that it's defaulting to the correct human reference sequence that was used by the laboratory when they classified your variant. And then if you have a question about how to input your particular variant into SIFT, which I find that I often forget, it's very helpful to look at this sample format link. They'll actually show you how to input it. So for this KCNQ1 variant, I told you I'd already pulled the genomic position, so I've gone ahead and pasted it in according to their recommended input. So you basically input your chromosome, genomic position. You have to indicate orientation, so either 1 or -1, representing whether you're on the forward or reverse strand. A quick way to tell that in this case is because we have the genomic position and we see the genomic position variant is represented as A to G, that corresponds to the variant change when we look at the cDNA A to G. So that means that we're on the forward strand, so you would use 1. But let's say this genomic position, this was actually the reverse of the cDNA, then that would indicate to us that we're on the reverse strand, and you would need to put -1 in your input here. And then you put the old nucleotide and the new nucleotide. Then if you scroll down, I like to ensure that I click this display multiple rows for multiple transcripts, and that's just because these different tools can sometimes give you different predictions according to different, for instance, KCNQ1 transcripts. So I want to make sure that I really can look, am I using the same transcript when generating my SIFT prediction as that of the lab and how they've written our variant. And then myself, I like to just include gene name and the ensemble gene ID just to make sure I can see that we're really looking at the KCNQ1 particular variant I've submitted, but you might choose to input more of these. So then you hit submit. It will process our submission. And then basically you would click here, proceed to SIFT results page, and then you can view or download the results. So I always just view them in my browser by clicking on that link. And as you can see here, because we had clicked that box of show us multiple transcript predictions, each line represents a different KCNQ1 transcript. So as you recall, the lab had given me, this was the RefSeq transcript, which is the major isoform of KCNQ1. So that is actually corresponding to this third row right here. So I want to know what is SIFT predicting for this particular transcript and my variant, which is right here. So I would go to this prediction column, and SIFT is calling this damaging. So the two possible outputs from SIFT in terms of a prediction are damaging or tolerated. So in this case, SIFT said damaging, which is in agreement with polyphen. So far we've queried 2 different in silico tools for the same variant, and they are in agreement that this variant is damaging, predicted to be damaging on the protein. Okay, so then let's take a look at the same variant in MutationTaster. So again, MutationTaster, if you Google search it or just type in this URL, you can get there quickly. And just a quick word about MutationTaster, it can give you missense predictions, just like SIFT and polyphen, but it also can incorporate insertions, deletions, nonsense variants, and splice variants. And you can actually input in both ways. You can either take the cDNA nomenclature that was provided to you by the laboratory, along with the gene name, or you can query by chromosome positions, like you just did with SIFT. And then it'll output a prediction of pathogenicity as well as the kind of summary as to why it generated that prediction, and then the probability as well. Okay, so let's first try it by the sequence cDNA nomenclature. So that would be this part right here, the C.dot nomenclature. So first you'd write in your gene name, KCNQ1, and then basically make sure if nothing auto-populates here to show you the different transcripts, click on this Show Available Transcripts. And so in this case, as I mentioned, you really want to make sure you're using the same transcript. In this case, that happens to be the major KCNQ1 isoform, but NM000218 is what we're looking for, and we see that right here. In most cases, that will correspond to the longest isoform, but not always, so it's important to make sure you're using the same one that the lab provided you. Then click coding sequence, and then because we're dealing with a single nucleotide variant, you would be in the second box right here. So it's asking you for your cDNA and then the new base involved in this single nucleotide variant. So I would put in 944, and then the new base is G for our particular variant. I'm going to do that, 944, and then the new base would be G. Then you go ahead and hit continue, and it gives you its prediction. So for that transcript of KCNQ1, it predicts that this missense variant is disease-causing or damaging on the protein. MutationTaster actually gives you a lot more information than just the prediction. So I'll walk you through a little bit about that. But I'm going to first just write disease-causing. So just briefly, MutationTaster, SIFT, and polyphen all were in agreement that this variant is damaging. So per the new ACMG guidelines, that would be, because they all are in agreement and use multiple tools, that would be sufficient to apply that piece of supporting evidence. So again, that can't be the only thing that you use for classifying a variant as pathogenic or benign, but that could be used as a piece of supportive evidence. So other information here, as I mentioned, it gives you the prediction at the top, either disease-causing or polymorphism if it thinks the variant is tolerated. It tries to give you a probability. I find their explanations and documentation pages very helpful if you want more information. And then they try to give you a summary as to what generated that prediction. So they'll basically say, this is a missense variant, which it's good to double check, did it actually query your particular variant? In this case, it did. Tyrosine is cysteine at position 315. And then it actually is able to tell you if there is another variant at that same position in either HGMD, the Human Gene Mutation Database, or ClinVar. So importantly, you should go actually look at those sources if, in this case, it's saying there is something at that position, because it could represent your exact variant that's been associated with the disease, or it could be saying there's another variant at the same location that's been reported, but it's actually not your exact same variant. In general, unless you have a splice variant that you know is affecting the canonical splice sequence, so either your splice acceptor or donor site, we find the splice site changes to not actually be accurate, and so would not really trust if you see this unless you know you're dealing with a canonical splice consensus sequence variant. And then as you scroll down, again, this documentation page explains all of these different fields, but you can see this links you to the dbSNP page, and then you can actually get direct links to HGMD to look at the variant described at that position. All right, so the other way that you could input the same variant is in the upper right-hand corner of the MutationTaster homepage. You would click Query Chromosomal Position. So just like SIFT, you could put in, if you have it, the genomic position. So in this case, chromosome 11, and then I'm going to highlight genomic position, and then you're going from an A to a G. So 11, position A to G. And then go ahead and hit Continue. And the only difference here is it's going to generate the prediction for each KCNQ1 transcript. So not, whereas previously we gave it the transcript we were interested in, this is going to generate the predicted effect of your variant on every KCNQ1 transcript. And it is being a little bit slow right here. Okay, so this is the result output if you've queried by a chromosomal position with a MutationTaster. So as I mentioned, each line shows you a different KCNQ1 transcript. Here's ours, the tyrosine 315 cysteine. And you see it's called disease, it's predicted to be disease-causing or damaging in both transcripts. Sometimes that will be different. And otherwise, it's the same information represented. Okay. All right, so in this case, as we already said, MutationTaster, SIFT, and polyphen were all in agreement that this particular missense variant is damaging, so you can use this as a piece of supporting evidence. And so I want to just take you through a couple more complex variants and how you input them into MutationTaster. As I mentioned, of the three I'm showing you today, that's the only one that you can really use for non-missense variants. Because polyphen and SIFT are only for missense variants. I'm going to go ahead and close out of this, clear my input. So the first one is a is a deletion, and so the way that you could query that in MutationTaster is on the home page. So again, here's the variant I want us to look at. It's MYBPC3, and then it's a CA deletion that leads to a frame shift. So a quick word in general, with frame shifting or premature truncating variants, in silico predictions are basically going to call it disease-causing just based on the fact that it's leading to a premature stop codon. So it's not really an independent kind of piece of supporting evidence there, but you can generate a prediction from MutationTaster for that type of variant. So the way you would do that in this case is you would, just like we did before, input the gene name, MYBPC3. Then you'll see the transcripts pop up. In this case, I already knew the isoform we were using is the 000256 ref-seq transcript. If I click on that one. And then here, because we're dealing with the deletion, you would actually use this bottom input box. And so here it asks for the last wild type or normal base before the start of your deletion, and then it asks for the first base after the deletion. So in that case, we have your coding sequence here, which is what we're looking at. So the last normal base before this deletion starts would be 3689, because you see it starts at 3690. So I would write in 3689. And then similarly, the last involved base is 3691, so I would write the normal one after that is 3692. And then if it was an insertion, which this is a deletion, so it's not applicable here, you would input what inserted bases were involved. But again, this is a deletion, so we're going to just hit continue. And then for that particular transcript of MYBPC3, it gives you the prediction. So again, as I mentioned, because this is a 2-base pair deletion, it leads to a frame shift, and therefore MutationTaster is calling it disease-causing just based on the fact that it'd be predicted to lead to nonsense-mediated decay. But you can just double-check, did they really capture the variant appropriately? Did I input everything appropriately? So phenylalanine 1230 leucine frame shift, and you see the same thing representing there, and the C dot correlates as well. So that's the way that you would input a deletion or an insertion. And again, not applicable for polyphen and SIFT because they can't take an insertion or deletion. Okay, so then finally, what if you wanted to look up a splice variant? So in this case, I have an MYBPC3 splice variant that affects the canonical splice sequence, this -1 of the canonical splice consensus. So I'm actually going to use I find it easier for this type of variant to use the query chromosomal position. So again, on the MutationTaster homepage, on the top right corner, it shows you this query chromosomal position. And I've already gone ahead and pulled it in this case. Again, if you have questions on how to get genomic position, I'd encourage you to check out that video. But basically, you would write in chromosome 11, copy your genomic position, and then C to T, so you would do reference allele C, new allele T, and then hit continue. So again, because you used chromosomal position, it's going to give you the predicted impact on every MYBPC3 transcript. In all of them, it's calling it disease-causing. And then you can scroll down and it'll show you each transcript and why they're predicting it to be disease-causing. So as I mentioned, unless you're dealing with a canonical splice variant, the splice site changes summary statement is not typically very accurate. But in this case, if you go down to the splice site section, because we know that we're actually dealing with the -1 of the splice acceptor, here it says acceptor lost instead of acceptor increased, sequence motif lost. So that's a way that you can, it's basically saying because this is affecting the canonical splice sequence, that would be predicted to be deleterious. Okay, so that is a very quick overview of 3 different in silico tools and how you use them in accordance with the new ACMG guidelines in terms of classifying a variant. Again, can't be the sole piece of evidence, but sometimes can be helpful if you find they all agree in a prediction of a particular variant. Again, I will point you to the ACMG guideline recommendation, which provides even more information. And if you have any questions, feel free to let us know. Thanks.